Hello everyone, here we are again with the new topic for today. We're still dealing with the metabolism of carbohydrates, but this time I'm gonna talk about the metabolism of some other carbohydrate molecules like fructose, galactose, mannose, lactose, and I'm gonna talk about uronic pathway in the end. Speaking of fructose molecule, it enters our organism through diet. So it's present in the form of saccharose in numerous fruits and sweets and industrial processed food, or in the free form, in the form of fructose, for example, from honey. If you remember our previous stories, we mentioned that fructose is absorbed from intestinal lumen by facilitated diffusion with the involvement of GLUT5 transporters. And we also emphasized the fact that comparing to galactose and glucose, absorption of fructose is relatively slow. Fructose can be further metabolized to give glycolytic intermediates, so it can be introduced to glycolytic pathway in several manners or using several mechanisms. So muscle, adipose tissue, and other tissues are catabolizing fructose in the way that phosphorylation of fructose occurs to fructose 6-phosphate, and this is completed by the enzyme hexokinase. The donor of the phosphate group is ATP molecule. Hexokinase is an enzyme which we know a lot about regarding the glycolytic pathway. And if you remember, I have mentioned that hexokinase is the enzyme phosphorylating hexoses. So it's not specific to glucose. Of course, it's the major substrate, but it can phosphorylate other hexoses as well. And now we see the confirmation of this fact. So hexokinase can phosphorylate fructose to fructose 6-phosphate, but this phosphorylation is rather slow, and hexokinase has a low affinity to fructose and other monosaccharides, but glucose, for which, if you know, it has a rather high affinity. So fructose 6-phosphate, which is formed by this phosphorylation, is now an intermediate of glycolytic pathway, so it can continue degradation in glycolytic steps like phosphorylation to fructose 1,6-D-phosphate and further degradation to DHAP and GAP. But anyway, in liver, and to a lesser extent in kidneys and intestinal mucosal cells, the situation is slightly different and it's kind of more complicated. Because if you remember, we stated that in the liver cells, instead of hexokinase, the major enzyme which phosphorylates glucose is glucokinase. Glucokinase is specific for glucose, and that is the name. The name glucokinase says itself. And we mentioned that the activity of hexokinase in liver is rather slow. So the catabolism of fructose and its introduction to glycolytic pathway cannot be completed by this pathway. So it undergoes more complex degradation, more complex catabolic pathway containing 7 enzymatic reactions. As a result of this pathway, fructose is converted to dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, both of them intermediates of glycolytic pathway. So this is the figure representing the catabolism of fructose in liver cells. So first step of this catabolism is phosphorylation of fructose to fructose 1-phosphate, catalyzed by enzyme fructokinase, which is at the same time the major kinase of this catabolic pathway. The donor of the phosphate group is ATP molecule, and this enzyme has a very high Vmax, maximum velocity, so this phosphorylation is rather rapid. So it occurs rapidly. In the next step, fructose 1-phosphate is degraded or hydrolyzed by the enzyme specific fructose 1-phosphate aldolase or aldolase B to dihydroxyacetone phosphate and glyceraldehyde. It is very well known that DHAP is already a member of glycolytic pathway, so by the action of triose phosphate isomerase, it's converted to glyceraldehyde 3-phosphate and further continues the glycolytic pathway. But what happens with glyceraldehyde? So from this point, we have two different options. There is an option to phosphorylate the glyceraldehyde directly by glyceraldehyde kinase, and again, the donor of phosphate group is ATP molecule. So glyceraldehyde is phosphorylated to glyceraldehyde 3-phosphate, thus involved in the glycolytic pathway. But on the other side, glyceraldehyde can be reduced to glycerol, so its aldehyde group can be converted to alcoholic group by alcohol dehydrogenase, and the donor of reducing equivalents is NADH coenzyme. Glycerol can further be phosphorylated, but only and exclusively in liver cells by glycerol kinase, and we are gonna discuss about this later on in lipid metabolism. So glycerol can be phosphorylated by ATP and glycerol kinase to glycerol 3-phosphate, and further on glycerol 3-phosphate can be oxidized by specific dehydrogenase, and the acceptor of the reducing equivalents is going to be NAD plus coenzyme. So in this reaction, glycerol 3-phosphate is going to be converted to DHAP, and thus it's the intermediate of glycolytic pathway. It can be isomerized by triose phosphate isomerase to glyceraldehyde 3-phosphate, which can continue the glycolytic pathway further on. So this is how fructose can be involved in glycolysis in liver cells. Speaking of enzymes which are important for this pathway, so those are fructokinase, as I've already mentioned, and aldolase B. I have already stated a couple of facts about fructokinase, but aldolase B is worth memorizing. Actually, it is one type of aldolase enzyme, so it is one of the isoenzymes of aldolase. Actually, aldolase exists in 4 isoenzyme forms: aldolase A, B, C, and fetal aldolase. Aldolase A is found in muscles and most other tissues. Aldolase B is specific for liver, renal tubule cells, and intestinal mucosal cells. Aldolase C is found in brain, and it's very similar to the fetal aldolase isoenzyme. All of those isoforms of aldolase actually are capable of hydrolyzing fructose 1,6-D-phosphate, as we learned normally in glycolytic pathway. But the specificity of aldolase B is that it can hydrolyze fructose 1-phosphate as well. Speaking of that, aldolase B is actually the regulatory or rate limiting enzyme of this pathway. Regarding its properties, we can say that it exhibits much lower affinity for fructose 1-phosphate than from fructose 1,6-D-phosphate. So at physiological levels, in normal conditions, it exhibits rather low activity, and this degradation and the fructose catabolism is rather slow. But fructose can also be synthesized in our organism in a specific pathway which is called polyol pathway. So fructose can be synthesized from glucose. The pathway is named according to the first step of this pathway, which yields the formation of sugar alcohols. So speaking about the glucose converting to fructose, in the first step, glucose is reduced. Its aldehyde group is reduced to alcoholic group by the enzyme aldose reductase, and sorbitol as polyol is produced. In the second step, sorbitol is oxidized to D-fructose by sorbitol dehydrogenase, and the acceptors of reducing equivalents are NAD coenzymes. This synthesis of fructose occurs in seminal vesicles, and it's important because this is the way to synthesize fructose for seminal fluid. In order to explain this, I have to tell you that spermatozoa use fructose as a major fuel source while in seminal fluids because it's the slow energy, it's low affinity energy, and it is enough just to maintain the status quo. But in the female reproductive tract, spermatozoa actually switch to glucose, which is like a turbo fuel, and increases the synthesis of ATP molecules. Why is this so important for seminal vesicles? Because it actually prevents acrosomal breakdown of plasma membranes while spermatozoa are still in the seminal fluid. Synthesis of fructose from glucose occurs in other tissues as well, but is still a question about the exact function of fructose in these tissues. Speaking about the disorder of fructose metabolism, definitely it's based on the deficiency of the rate-limiting enzymes of its pathway. So the first one is fructokinase, and deficiency of fructokinase yields the formation and development of essential fructosuria. It's a rare genetic banning disorder. In this case, because of the deficiency of fructokinase, fructose cannot be phosphorylated to fructose 1-phosphate by this enzyme. But the good thing is that there is no accumulation of toxic metabolites of fructose. Fructose itself is not considered a toxic molecule. And on the other hand, there are other tissues and hexokinases that can slowly but can phosphorylate fructose. So this disorder goes almost nearly asymptomatic in the people carrying it. Speaking of the fructose taken from the diet, it can be phosphorylated by hexokinases in other tissues. So the metabolism is slow. Fructose can appear in urine because there is no renal threshold for fructose, and unlike glucose, it is not required to have a high concentration of fructose in blood in order to appear in urine. For fructose is just normal, and the disorder is fructosuria. Another enzyme which was rate limiting and regulatory is aldolase B, and its deficiency yields in the development of hereditary fructose intolerance, which is much more serious disorder than the previous one, with more severe consequences and symptoms. And the deficiency of this enzyme actually yields the accumulation of fructose 1-phosphate in liver and in kidneys. And as a result of this, hypoglycemia occurs. And the reason why hypoglycemia occurs, I need to remind you about the regulation of other metabolic pathways of sugars. When we mentioned that the fructose 1-phosphate inhibits glycogenolysis, I mean the enzyme glycogen phosphorylase. It also inhibits gluconeogenesis, and this is going to be the next video. Fructose 1-phosphate also inhibits aldolase activity. So as a result of it, it inhibits gluconeogenesis, I mean the usage of a lactate from anaerobic glycolysis, which yields the accumulation of lactate and lactic acidosis. Also, fructose 1-phosphate depletes the intracellular phosphate tools, because if you remember, I told you that fructokinase has a high Vmax. So the phosphorylation of fructose is rather rapid in this case, and it uses a lot of ATP, which is converted to AMP, which can be deaminated to inosine monophosphate, and as a nitrogen base it has hypoxanthine, if you remember our practical exercise, and it's converted to uric acid, so you have one more adverse effect of this accumulation. Also, the rapid loss of ATP from cells may yield in those extreme situations to the cell death. This fructose intolerance is more pronounced by its symptoms in infants and children, because in adult persons they already realized what's going on, so they can avoid it in a fructose intake in diet. But in infants and in children, it's manifested by poor feeding, by vomiting immediately after breastfeeding, by numerous intestinal discomforts, by failure to thrive, and so on. And prolonged ingestion of fructose may lead to hepatic and renal failure, which is a really, really severe consequence, which actually makes the usage of fructose as a substitution for glucose and saccharose rather questionable due to the over accumulation of those toxic metabolites in the form of fructose 1-phosphate. When fructose is used as a substitution in those intravenous infusions or enteral tube feeding. And now if you got so excited about what about so many fructose products on the market where fructose is used instead of glucose as a sweetener, oral ingestion of fructose yields to a rather slow absorption. So fructose is slowly absorbed, not a high concentration in blood, so it can be distributed to other tissues and slowly phosphorylated by hexokinase. So fructose ingested orally, it's not that problematic as taken on those mentioned other ways in cases with those fructose intolerance. And finally, the last regulatory enzyme regarding fructose synthesis is aldose reductase, which yields to the accumulation of sorbitol and galactitol from galactose reduction. And it occurs in patients with hyperglycemia or diabetes mellitus, and ends up in the formation of cataract. So when the concentration of glucose is high in blood, it increases the synthesis of sorbitol and fructose, and both of those are osmotically active molecules, so as a result you're going to have an increased osmotic pressure. Apart from that, you have the glycosylation of proteins, which yields to the formation of that characteristic opaque cloudiness of the lens. Additionally, apart from this, accumulation of sorbitol in nervous tissue in diabetic or hyperglycemic patients yields to the formation of very characteristic peripheral neuropathy, characteristic of patients with poorly controlled diabetes. Galactose. What we know about galactose is that it's 4 epimer of glucose. So what we need to do is to epimerize galactose at position 4 to glucose and to bring glucose to glycolytic pathway. So it looks so simple and easy, but it's not. So it's not just a simple epimerization at position 4, but rather it involves 4 enzymes for conversion of galactose to glucose 6-phosphate. This pathway is moderately active in adult persons, but in infants is even more pronounced and even more active due to the more portion of lactose in diet. So what happens in this pathway? If you take a look at the figure, galactose is first phosphorylated to galactose 1-phosphate by galactokinase. And further on, galactose 1-phosphate undergoes one very interesting transformation, which means the transfer of galactose molecule to UDP part of the molecule, forming UDP-galactose. UDP-glucose is used for this reaction, and the enzyme is galactose 1-phosphate uridylyl transferase. Glucose is released in the form of glucose 1-phosphate, which is converted to glucose 6-phosphate by phosphoglucone isomerase, and glucose 6-phosphate can be introduced to glycolysis or converted to glucose in liver. UDP-galactose is then, when activated, is then epimerized by specific epimerase at position number 4, which requires NAD plus as an intermediate to UDP-glucose, which can accept another molecule of galactose 1-phosphate. Regarding the disorders of this pathway, it's the deficiency of galactose 1-phosphate uridylyl transferase yielding the development of classical galactosemia. As a result of the improper function of this enzyme, accumulation of galactose 1-phosphate in liver occur, which actually affects the hepatic glycogen metabolism in the terms of its inhibition, and it also inhibits other pathways that require UDP sugars, and there are many of them. I'm going to mention some of them in a couple of minutes. So as a result of insufficient catabolism of galactose or suppressed galactose metabolism, the concentration of galactose rises in blood, and that yields to an increased reduction of galactose to galactitol in lens, which actually again results in the formation of cataract. Also, this galactosemia is manifested by an irreversible mental retardation, which requires an immediate diagnosis, which is accomplished and done immediately after childbirth. If prolonged, this galactosemia can derange liver function, may lead to the formation of jaundice, hepatomegaly, cataract, maybe even in some cases accentuated by severe hemolysis. And speaking of the management of this, elimination of galactose from the diet once the deficiency is diagnosed. Mannose is C2 epimer of glucose, and it's a component of some glycoprotein molecules in our organism. The catabolism of this monosaccharide is accomplished by its phosphorylation to mannose 6-phosphate by hexokinase, like other hexoses. And then mannose 6-phosphate is isomerized to fructose 6-phosphate by phosphomannose isomerase, and fructose 6-phosphate is then introduced in glycolytic pathways. So it's pretty easy to catabolize mannose molecules. But lactose is a molecule that can be synthesized in our organism, not only digested and degraded in gastrointestinal tract, but rather synthesized in our organism from UDP-galactose, the one which is activated, we saw it previously, and glucose molecules. Notice that in this case, for the lactose synthesis, galactose is not required in the diet because glucose can be converted to galactose, and actually that is the first step of this synthesis. So UDP-glucose, activated glucose, can be converted to UDP-galactose by the action of that specific 4 epimerase and upon the involvement of NAD as an intermediate. So lactose synthesis occurs only in lactating mammary glands of adult females during the lactating period only, and the enzyme which completes the synthesis is called lactose synthase. This enzyme has actually 2 domains. The first one exhibits enzymatic activity, and it's galactosyl transferase activity, which actually is responsible for the formation of beta-1,4 glycosidic bond between glucose and galactose molecule. The second domain is one modifier protein, alpha-lactalbumin, which is synthesized immediately after childbirth upon the action and signaling of prolactin hormone. So in the absence of prolactin, this molecule, this protein is not going to be synthesized, and the lactose synthase is going to be, activity is going to be inhibited. Speaking of UDP-glucose, we have mentioned it several times in glycogen metabolism, now in monosaccharides metabolism. So we know that UDP-glucose is synthesized from glucose 1-phosphate, as seen in this figure, in the reaction with UTP. And so far we know that UDP-glucose is involved and necessary for the biosynthesis of glycogen. Also, we know that UDP-glucose is needed for the biosynthesis of lactose. But UDP-glucose is a precursor for some other events in our organism. It's needed for the biosynthesis of proteoglycans, glycoproteins, glycolipids, but one of the major routes of UDP-glucose metabolism is the formation of UDP-glucuronate. UDP-glucuronate is a very important precursor for numerous events in our body, and I have provided you with a couple of examples here in a figure. So UDP-glucuronate is needed for glucuronidation or conjugation of bilirubin and its safe excretion from the organism. It's also used for binding OH group of numerous steroids, drugs, xenobiotics in order to form glucuronides, soluble molecules safe for excretion from the body. It's used for the synthesis of UDP-xylose, which is one of the components of glycosaminoglycans, also iduronate formation. So how UDP-glucuronate is synthesized? Actually, the main chemical change occurs at the primary OH group at the 6th carbon atom of glucose, which is oxidized to carboxylic group. This oxidation is catalyzed by UDP-glucose dehydrogenase, which requires NAD coenzyme as an acceptor of reducing equivalents. Certain small amounts of glucuronate can be obtained from diet or by chemical modification of inositol. The major function of UDP-glucuronate is binding to OH group of numerous molecules containing it, thus increasing its solubility. The major portion of UDP-glucuronate are found in liver, and the process of binding of UDP-glucuronate to OH group of those molecules is called conjugation, which is the major pathway of detoxification processes in the liver. A more detailed story about detoxification in some other videos. Catabolism of UDP-glucuronate goes in the way that the final product form is xylulose 5-phosphate, which can be further catabolized or modified in mammals, but not humans, to vitamin C. And instead of conclusion for this topic, I'm going to give you one fact. Apart from all those symptoms connected to galactosemia, accumulation of galactose 1-phosphate also inhibits the activity of phosphoglucomutase, which is responsible for interconversion of glucose 6-phosphate to 1-phosphate and back. So can you connect this fact with hypoglycemia and jaundice that accompany galactosemia? Take a few minutes to think about it and find the possible mechanisms and answers by getting back to what we've mentioned previously. You can pause the video as well, and I'm going to explain you which are the mechanisms for development of hyperglycemia and jaundice in this case. So let's start with hypoglycemia. Actually, the accumulation of galactose 1-phosphate, galactosemia in general, interferes with the formation of UDP-glucose. That is what we've seen. And it also interferes with the degradation of glycogen to glucose 6-phosphate. This is to be explained. If you remember my story on glycogen metabolism, we mentioned that 90% of glycogen is degraded to glucose 1-phosphate. Because of the fact inhibition of phosphoglucomutase, glucose 1-phosphate cannot be converted to glucose 6-phosphate. And there it stops. So in liver, glucose 6-phosphate from glycogen cannot be glucose 1-phosphate from glycogen cannot be converted to glucose 6-phosphate, cannot give glucose, cannot be exported to bloodstream. Only 10% of glycogen is converted to free glucose, which can be exported to bloodstream, but it's everything but sufficient to fulfill the pool of glucose in blood, so hypoglycemia develops. Speaking of jaundice, we mentioned that galactosemia interferes severely with the production of UDP-GLUCOSE. So the levels of UDP-GLUCOSE drops down because for UDP-GLUCOSE synthesis we need glucose 1-phosphate. And glucose 1-phosphate is obtained from glucose 6-phosphate. So if phosphoglucomutase is inhibited by galactose, accumulation of galactose, we cannot get glucose 1-phosphate, and we have to stop the formation of UDP-GLUCOSE. If we don't have UDP-GLUCOSE, you see a couple of seconds ago, we cannot synthesize UDP-glucuronate. If we don't have UDP-glucuronate in sufficient amounts, you cannot conjugate bilirubin to bilirubin diglucuronide. And as a result of that, bilirubin is accumulated in tissues manifested by the development of jaundice. So if you take a look at these metabolisms, despite the fact that they are sometimes marginalized in the favor of emphasizing the role of glucose in the overall carbohydrate metabolism, but other hexoses and other sugars are also very important in the proper maintenance of carbohydrate metabolism. So that would be the end of today's video and the end of today's topic on the metabolism of other carbohydrates. We have one topic left, which is going to be involved in the last video, gluconeogenesis, and by that we're going to close the story on carbohydrate metabolism, and we're going to move to something else. But until then, until gluconeogenesis, stay safe, stay healthy, be clever, be beautiful, and bye-bye. Looking forward to hearing you again. Bye-bye.